Hi, College Biology. This is your Chapter 9 set of lecture notes. And we are going to be working with the concept of going from genes to proteins. The central dogma is that messages will travel from the nucleus in the form of DNA. It will get changed into RNA, and then RNA will change into proteins, and then proteins control the reactions in your cell. The process of going from DNA to RNA is called transcription, and then the process of going from RNA to protein is translation. In this lovely picture to your right, you have a picture of everything that is going on. Hopefully you can kind of identify a few things that are happening here, even though you can't tell the whole picture just yet. You've got your DNA, which is your blue line here, and then you've got your RNA, which is this lovely red ribbon. You've got the nucleus, which is this purple. Notice it's a double membrane, and these pores in your nucleus allow the RNA to leave. You've got ribosomes here. You've got purple little dots and squiggles and stuff. These are your little amino acids that are going to be joined together, making a polypeptide chain and being carried in by tRNA, which are these blue things. And these parts down here are called codons and anticodons that join to one another. So I know all of that didn't make sense, but by the end of these videos, my goal is that if I would show you this picture on the test and be like, describe the 3 steps of transcription and translation using the diagram above, you would be able to do all of that. So without further ado, let's just begin in transcription. Transcription is the synthesis of an RNA transcript. So this little section here from DNA moving to single-stranded RNA is what's going on. You can also see that picture happening here, where RNA is the red and DNA is the blue. So how does that work? RNA polymerase, which is this fancy looking like gray, whatever it's called down here, it's an enzyme that's going to attach to DNA, and it's going to allow the DNA to be transcribed. Your template DNA is going to be going from the 3' to the 5'. So if you remember, your houses here are going to be upside down. This is your deoxyribose sugar. However, RNA, and if you remember Thursday, Friday group, the sugars always stack like this. Anytime they're being added to one another, you stack the base on top of the roof. But RNA goes backwards. So your ribose sugar is here, and it's going to attach in the 5' to the 3' direction, which is basically opposite of what the DNA does. So RNA polymerase will use a single-stranded DNA to synthesize a complementary, with the E, not the I in the middle of the word, complementary strand of RNA. Complementary with E means complete. I means like a compliment to you, like, oh, you look so nice today. RNA polymerase is not complementing the DNA in the fact that it's telling DNA it's pretty. It is completing the DNA strand as the complement on the other side of it. And it will build an RNA strand again in the 5 to 3 direction, which again is backwards from what DNA replication happens. So that's what you need to know about RNA polymerase. How does this work? So let's talk about the steps of transcription. The first step is called initiation. And initiation, what I was talking about is what happens. RNA polymerase will bind to a sequence of DNA called the promoter, and it is found near the beginning of a gene. Remember, we are transcribing genes. So these sites here in this picture, these green, are the promoter, and that's where the RNA polymerase will begin binding. And it will cause it to split, just like DNA helicase did in our DNA replication. So it will cause it to split, but it doesn't really begin transcribing until it gets to this yellow area of the gene. So that's kind of handy. It binds early and splits it, and then when it gets through to the area of transcription, that's when it will start building its gene, because you don't need more transcribed than what is part of the gene. The reason for that is what's being transcribed is going to translate to amino acids, and amino acids build proteins. So if you end up with extra amino acids, your gene just isn't going to work right. It's not going to work at all, probably, because it has to start with a very specific amino acid. So when it gets to this first set of 3 here, it would be, let's see, T, A, C is actually where it would always begin in eukaryotic cells. And prokaryotic cells, it doesn't always begin there, but in eukaryotic cells, it does. Archaeobacteria will always start there as well, by the way. It's a special case of prokaryotic cells. Anyway, I digress. Moving on. Each blank or group of co-transcribed genes So each gene is what that means, or a group of co-transcribed genes in bacteria. So bacteria DNA is so small it transcribes a whole bunch of genes at once. They all go together, but in eukaryotic cells, genes are separated. So each individual gene or group of them has its own promoter. So there will be promoters scattered throughout your DNA every time a gene would need transcribed. Once bound, RNA polymerase will separate the DNA strands, providing the single-stranded template needed for a transcription. So the initiation, basically, you bind to the promoter region and you separate it. That's all it is. That's all that was meant to say. The second step, elongation. What do you think is happening? The RNA strand is going to elongate, or we're going to build it. So one strand of DNA, or the template strand, acts as a template for the RNA polymerase. As it reads the template one base at a time, the polymerase will bind an RNA molecule out of complementary nucleotides, making a chain that grows from 5 to 3. Number 3, the RNA transcript carries the information as the non-template or coding strand of the DNA, but it contains the base uracil instead of thymine. Uracil is URACIL instead of thymine, so that's what that U is. Like I said, it starts transcribing at this TAC, but instead of it being this complementary T, it will be AUG is the complementary strand of it. The coding strand is the opposite end, so it's going to mimic the coding strand with the exception of the T's or the U's. When you do your assignment in class, you'll actually see this and you'll be transcribing, but anytime you see a T, you will change it to U. So elongation is just the building of that RNA strand. That's all it is. Okay, the last step is termination. So what do you think termination is? Termination is when it stops. So in termination, sequences called terminators signal that the RNA transcript is complete. Once they are transcribed, see this terminator sequence here. And once the RNA sequence is transcribed, the transcript, they cause the transcript, I'm stuttering, to be released from the RNA polymerase, and you can see this happening here. So it kind of causes, the terminators kind of cause it to fall off of itself as it pulls away from the RNA polymerase. The RNA polymerase will go transcribe the next gene, and then your RNA single strand here is almost complete. In bacteria, it is complete, but it is not complete in eukaryotic cells, so it has to go through additional stuff. It's not quite done yet. So eukaryotic RNA has to be modified. So RNA modifications is your next words. And what does that modifying? Wow, I've lost my spot on my teacher notes. I'm sorry. I always try to follow along here so I can keep it nice and neat. So again, let me just go through the teacher notes that I have separate. By the way, I got all these from Khan Academy. All the pictures are from Khan Academy, so I got to give credit where it's due. I did not rewrite the words or anything. In bacteria, RNA transcripts can act as messenger RNAs right away, but in eukaryotes, a transcript of a protein coding gene is called pre-RNA, and it must go through extra processing. So this top part is called RNA processing. This is where we modify the transcript to form mRNA. So it's not quite yet. The transcript is rough. So eukaryotic mRNAs will undergo what is called splicing. In this process, parts of pre-RNA called introns are chopped out. So guys, it's really funny. You would think introns are what is kept in and exons are what exits, but that's actually the opposite. Introns are what gets spliced out, and then the exons get joined together. So in this example, you've got introns that are marked here and here. They get spliced, and your mature RNA gets sealed together. But that's still not the last thing that happens. In modifications, increase the stability of mRNA splicing and gives mRNA its correct sequence. If the introns are not removed, they'll be translated along with the exons, producing a gibberish polypeptide. So you have to have these introns out. And the other thing that has to happen is you got to put things on there. So the last part that has to be added on, and I don't know what happened to my notes here, but one of the things that has to happen is you have to have a poly A tail be put on the end of it. That's very important. That kind of keeps it stable there. And you also have to have a 5' cap put on it. Oh, it's right here. Pre-RNA ends are modified by the addition of a 5' cap, which is your G, and a 3' poly A tail. That little like quote type mark there is called prime. So a 5' cap and a 3' poly A tail. So those things keep it stable and make it work. All right. In the next one, we will talk about translation, but just to sum it up, you have initiation, elongation, and termination. Initiation, RNA polymerase will bind and separate the DNA. Elongation, just the RNA gets built. You add in the complementary bases. Remember, U replaces T. And then termination is when the process stops and it gets released by the RNA polymerase. And then finally, it has to be modified in eukaryotes, which means introns are spliced out and you add on your 5' cap and your poly A tail. And now it's finally ready to leave the nucleus and begin translation. And that'll be in your next video.